Okay, good afternoon. Let's go ahead and start here. So today is June the 2nd, a Wednesday, and let's go ahead and talk about some of the things that I've mentioned over the past 24 hours or so. Uh, and most of that has to do with your lab work. So hopefully by now things are starting to settle down. Uh, and some of you had expressed that you did not really know or were not aware of the requirement for getting the lab manual. It was basically mentioned in the other emails that I sent out earlier, but it's understandable if things fall through the cracks because of so much going on at the same time. So as I mentioned, as for now, uh, almost everyone has Connect access. So at least finish those Connect virtual labs that I posted, right? And as you get your lab manuals coming in, please make sure that you catch up on the work and turn that in as well. So I'm just gonna wait till the weekend and then take a look at the work and then start grading all together, okay? Um, and so I have extended the deadline for students who are facing delays getting their stuff in. Okay, also I'm almost done with your study guide for your first lecture exam, which seems like is on this coming Tuesday, and I should be sending that out uh, if not today, then hopefully by tomorrow for sure. So that way you guys have a good amount of head start preparing for that. Also make sure you are listening to the recorded lectures that I will continue to send out and the notes and take your own notes as well. I sent out a study group link that Juan from our class graciously shared with me, as I mentioned. Feel free to join that. It's always a good idea to work as a group or um, have partners who can quiz each other and then basically bounce off ideas off of each other, as I mentioned before. Okay, so with those words of reminders and things that are coming up, uh, any other questions that might pop up, please do not hesitate to let me know. Send me a quick email and I'll be more than happy to answer those. So looking back at the schedule, we are here at chapter number 2, and this is what we're gonna go over today. The chapter here basically is concerned with the chemical basis of life. So hopefully, as you remember from before, there are different levels: chemical, then cellular, then tissue, organ system, and then organism. So we're going to go in that order. All right. So let's start with the very basic level, the chemical level. So here's a lot of high school chemistry, chemistry 101, if you will, that is involved. Things like matter. All right. So everything is made up of matter, whether it's a solid, liquid, or a gas. And matter is defined as anything that has mass and occupies space, right? Whether again it's solid, liquid, or gas. So what types of matters are found within the human organism? All three types, as you can see here: solids, liquids, and gases, right? Some examples of solid matter, well, we all know that: muscles, bones, blood cells, cells in general, right? These are all solid types of matter inside the human body. There's liquid, blood, plasma, lymph, synovial fluid, serous fluids. All those are examples of liquid matter. And then gases, dissolved gases like oxygen and carbon dioxide, which are basically circulating in our bloodstream at any given time, right? So the basis of all matter is an atom. Atom is the smallest particle of an element, the smallest unit of an element, just like cell is the smallest structural and functional unit of life, okay? Uh, Atoms make everything: gases, solids, and liquids, right? And so far in the periodic table, which looks like this, what we have uh, is these elements which are arranged in a certain order, right? So for example, you have rows, as you can see, okay, rows here, and then also columns and rows, and 92 naturally occurring elements is what you see here, all right? So the question arises, how do we even put these elements in order uh, based on what, right? So if you pay attention to this little number at the top, so the symbol for hydrogen is H. So these are symbols for these elements: hydrogen, helium, right? Lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, so on and so forth. So these are symbols, right? Most of them have to do with the name of the element. Some of them are derived from ancient Latin, Greek root terms. Regardless of that, if you look at the number at the top, it's increasing by one. So one, then two, three, four, five, six, seven, eight, nine, 10, 11, 12, and on it goes. All right. So that number is called the atomic number. That's what it's called. And the atomic number is the number of protons that a given atom has. Okay. Now, at the same time, you also see these numbers at the bottom, right? So 1.008 for hydrogen, 4.003 for helium, 6.941 for lithium, 9.012 for beryllium, so on and so forth. So that is the mass number. And the mass number basically is the number of protons plus neutrons in an atom. So what are these particles anyway? Protons, neutrons, electrons, okay? So that's what we're gonna look at here shortly. So these are subatomic particles that make up an atom. They're found inside of an atom and in a certain arrangement, okay? So the first thing to um, understand when looking at these numbers is, well, know that the top number, which increases by one, is the atomic number, which is the total number of protons. And then these bottom numbers here are the atomic mass numbers, which are the numbers of protons plus neutrons. Okay, that much we have to remember. And then, of course, the symbols uh, of the elements as we see here. Okay, so here's a list of some of the common elements which are found inside of the human body. As you see, they're divided into major and minor or trace elements. Okay, so what's the difference? Major elements are found in greater numbers, of course. About 99% of your body weight is composed of these elements, and minor elements are responsible for less than 1% of your body weight. Okay, so what are some examples? If you look at the major elements here, uh, so if you remember the acronym CHOMP, CHONP, right? CHOMP, uh, that will help you remember what are the major elements found in the human organism, right? CHOMP, C for carbon, H for hydrogen, O for oxygen, N for nitrogen, and P for phosphorus. You see all of them, right? Uh, or you can add a C there also, so CHONC or something like that. So that C might be for calcium as well, all right? So these are the major elements, right? And then the minor ones are there: sulfur, potassium, sodium, chlorine, magnesium, and iron, okay? Interestingly, iron is also one of the minor elements, even though you might think, well, we need iron for forming blood, for hemoglobin, which is the red pigment in blood and all that. Even so, iron is one of the minor elements, okay? As is sulfur. Okay, so moving on from there. Okay, I don't know why it's looking all funky here. All right, so these are the three subatomic particles that I mentioned, right? Protons, electrons, and neutrons. That's what we are looking at here. And so what are some of the properties of each of these subatomic particles? Protons, they start with the letter P. Think of P for positive or plus. Protons are positively charged particles. They have a positive plus sign on them, positive charge, right? They also have some mass, and their mass is about one atomic mass unit, 1 AMU. Okay, that's what there is to remember for protons. Neutrons, on the other hand, look at the word itself. Neutrons, they're neutral. They're not positively or negatively charged. They're neutral, but they do have mass of about the same magnitude as the proton, which is one atomic mass unit again. So they both have some mass, protons and neutrons, one atomic mass unit. Protons are positively charged, neutrons are neutral. Finally, we're left with electrons. These are very tiny particles, extremely small particles, only, as you can see here, 1/1800th of the mass of a proton or neutron, negligible mass. So no mass literally for electrons, but they do have a charge, and that's negative. So electrons are tiny, minute, minuscule, negatively charged subatomic particles. We talked about these earlier, chemical symbols. Each element has a symbol that has to do with its name, either modern name or ancient Latin/Greek derived name. Atomic number is the total number of protons. Uh, Average atomic mass is the mass of both protons and neutrons, because remember, electrons literally have no mass almost. So protons and neutrons uh, are the two particles that give you the average atomic mass of an element, okay? So So how do you calculate the number of different particles in an atom? So again, atomic number is the same as proton number. We know that, right? So what happens when you take mass number and you subtract proton number or atomic number from it? Then you get the number of neutrons. Makes sense, right? Proton numbers are atomic numbers. Mass numbers are both protons and neutrons. So if you subtract uh, the atomic number from the mass number, you get the number of neutrons. So that's some basic math right there. Okay, now, so how are atoms constructed? Here's a picture. This is a model of a typical atom, okay? So one thing to observe here is you have something, a ball of these spherical particles in the middle, and then some tiny spherical uh, particles going around. It's almost like the planets orbiting the sun, right? So what you have in the center here is called the nucleus of the atom, not the nucleus of the cell. That's a different type of nucleus in cellular living things. This is the nucleus of the atom. And you can see two colors here, blue and green. So two different particles, which are neutrons and protons. Neutrons and protons are compacted together in the center of an atom in the nucleus. These yellow subatomic particles that you see going around the nucleus, those are electrons. The yellow ones are electrons, which are far smaller, almost no mass, negligible mass, but they do have a charge. These are negatively charged. The protons are positively charged. The neutrons have no charge whatsoever, okay? So since the protons, let's say green is protons, is it? Yes. So the green ones are protons. So they have a positive charge. Electrons have a negative charge. We know opposites attract. So these positively charged protons keep the negatively charged electrons uh, spinning around them, right, in orbits. And this is basically what keeps them in an orbit, this attraction, all right? Um, so this is a typical configuration of an atom, as you can see here. Now, each of these orbits that the electrons are revolving in around the nucleus, each of them is called a shell, okay? And uh, the farther away that you move from the nucleus in the orbits, the lesser the amount of energy that these electrons have. Because the farther they move from the nucleus, the lesser the attraction they have towards the nucleus, right? So they're easier to knock out of their orbit if they're farther away. The ones which are in the closest shell, they have the most energy because they are closest to the nucleus. So there's most attraction there, okay? So the innermost shell basically has two electrons in a typical atom, as you can see here. The second shell can have up to 8 electrons, okay? And the shells which are close to the nucleus must be filled first by the electrons. That's how they line up. They first go in the first shell, right, when they're attracted, then the second shell gets filled up, and so on and so forth. One thing that would be good to remember at this point is all atoms rather love to have 8 electrons in their outermost shells. That's just how it works for atoms. They like having 8 electrons in their outermost shell. The outermost shell is also called the valence shell, the valence shell. And the electrons which are in the outermost shell are also called valence electrons. And they like to have 8 of those. That's called the octet rule. Oct means like 8. So octet rule, they like to have 8. Okay. Next up, we are looking at something called isotopes. What does that mean? Iso means same. Iso, the prefix iso is same. Topes for topography or topy, shape. So same shape is what isotopes literally means. But what is an isotope? If you take an element, let's say carbon here, right? It's the same element, but you can see that carbon exists in at least 3 different forms, okay? So one is called carbon 12 here, one is called carbon 13, one is called carbon 14. So what's the difference? And they're all carbon, so how are they different? Why these different numbers? Because if you look in the first one, this type of carbon atom has 6 protons, you can count them, the green ones, 6 neutrons, the blue ones, and then 6 electrons altogether. You can count the electrons too, there are 6. So 6, 6, and 6. So when you add up the number of electrons and protons, 6 and 6, 12. So it's called carbon 12. But carbon can also occur in a different arrangement, which is called carbon 13. Here, you have 6 protons and 6 electrons again, but you have 7 neutrons. You have one extra neutron. So 7 plus 6 gives you 13, okay? So one number more as far as neutrons are concerned. So this is carbon 13, still carbon, but different number of neutrons here. So then you go to carbon 14. What do we see here? Again, 6 electrons and 6 protons. That doesn't change. Electrons and protons, same number, because protons are positive, electrons are negative. They kind of neutralize each other, nullify each other, right? So that doesn't change. But look here, look at the number of neutrons. Here you have 8 neutrons, 2 extra neutrons. So 8 plus 6, 8 neutrons plus 6 electrons, 14. That gives you carbon 14, all right? So the same element can occur in different types of isotopes, which isotopes are the same element in different forms, which have the same number of protons and neutrons, but a different number of neutrons. That's what you have to remember, okay? So why does it matter anyway? Well, it matters because it has certain clinical applications, and also if you're a paleontologist, right? Paleontologists are scientists who study fossils, and one of their major jobs is to date fossils and rocks in which those fossils are found, right? How old, like dinosaurs, they went extinct 65 years old, 65 million years back. So how do the paleontologists know that? So they do something called carbon dating, literally. That's what that's what it's called, carbon dating. So how that works is basically carbon exists in all 3 forms, but the most stable form is this one, carbon 12. That's the original. So these carbon 13 and carbon 14, they lose those extra neutrons over millions of years to turn into carbon 12, okay? And each element has a certain given number of set years during which uh, those neutrons basically are ejected. So that, let's say, it takes 5 million years for carbon 14 to lose that single neutron, get rid of it, and turn into carbon 13. And then it takes another 10 million years for carbon 13 to get rid of that one neutron and turn into carbon 12. So if you see a fossil or a place or a rock where uh, you have 100% carbon 12 or let's say 50% carbon 13 and 50% carbon 12, you can basically calculate the amount of years that have passed just based on uh, what is the amount of these 2 isotopes in relation to each other, okay? So what are some medical applications of these isotopes? So here we are talking about radioisotopes. What are these? Radioisotopes are, again, different forms of the same element, and when let's say, for example, iodine 12 and iodine 13. When iodine 13 turns into iodine 12, meaning it gets rid of one neutron, that neutron basically glows in the dark, if you will, when it goes out. So it acts like a tracer bullet. You can literally see it glow in the dark. So these radioisotopes are used to basically light up the human body. So you drink some iodine 13, and they don't always use iodine. They use different elements which have a very short half-life, meaning they're always shooting out neutrons, very little short half-life. And so when one type of uh, radioisotope breaks into the other type, it releases these glow-in-the-dark neutrons, and so it lights up the human body. It allows you to see different tissues because they light up, okay? So uh, this is what radioisotopes are. And what types of radiation do they give out? Alpha and beta particles and gamma rays. Alpha particles are the least penetrative. They don't penetrate too much. They cannot go across like a thin sheet of paper, as an example. Beta particles are slightly more penetrating. They can go through like a block of concrete, as an example. Gamma rays are the most penetrating. They can go all across. Gamma rays are those penetrating rays that can give you cancer. So if you're exposed to higher levels of gamma rays, if you work in a nuclear facility or nuclear medicine or something, it can literally damage DNA in your cells and cause mutations and cause cancer. Interestingly, gamma rays are also used during radiotherapy to burn and destroy those cancer cells, okay? So that's basically what half-life is. Physical half-life, the time for 50% of a radioisotope to become stable. For example, for 50% of carbon 13 to break down into carbon 12 can happen from a few hours to thousands of years, depending on what type of an element you're looking at. So then what is biological half-life? The time required for half of the radioactive material from a test to be eliminated from the body, okay? So as we see here during different medical procedures, and you will see this if you work in nuclear medicine, right? Nuclear medicine is the branch of medicine where you deal with imaging techniques, CT scans, X-rays, so on and so forth, also radiotherapy for burning out off cancer cells, right? So uh, here you can literally use these unstable radioisotopes as tracer bullets, as glow in the dark kind of things, all right? And one example is for thyroid studies. So let's say someone has hyperthyroidism, okay? What does that mean? The thyroid gland, which is this butterfly shaped gland in your neck, it secretes a number of hormones. Two of the hormones are T3 and T4. T3 for triiodothyronine, T4 for thyroxine, right? So what these two hormones do is they regulate your metabolic rate, your metabolism, how fast you burn calories, how hot you feel, or how cold for that matter, how easily you put on weight. All of that is a factor of your thyroid gland, right? And women are more prone to hypothyroid conditions, right? Or thyroid conditions in general, right? So women uh, more prone to thyroid issues, right? And why is that? Because women in general, well, they get pregnant, right? And during pregnancy, the thyroid hormone has to pump out double the amount of hormones because it has to up your metabolic rate to sustain the growing embryo within your womb, within the uterus, right? So you're literally pumping out hormones for two. Sometimes you have triplets or who knows, quadruplets, right? And so your thyroid would be a whole lot more active. So that causes issues. Women are also more prone to autoimmune disorders, right? Their own immune system going haywire and attacking their own tissues, right? So why are women more prone to autoimmune disorders? Same thing. They get pregnant and the pregnancy sometimes gives rise to a blood mismatch. So the mother might be Rh negative blood type, which we will talk about later in more detail. The fetus or the embryo that is developing in her uterus might be an Rh positive child, right? And so this blood mismatch causes the immune system to go a little haywire, if you will, right? And we'll explain what we mean by that, right? Sperm or semen from the male partner itself is kind of like an allergen, an antigen. It can cause the woman's or the female's body to pump out antibodies even against sperm and semen of the male, right? So pregnancy and then the presence of sperm itself, uh, and also estrogen, the female sex hormone, seems to ramp up your immune system. So all of these factors come together to to conspire against women for having a disproportionately greater number of autoimmune diseases, right? Like lupus, SLE, systemic lupus erythematosus, like rheumatoid arthritis, like autoimmune thyroid problems, thyroiditis, even thyroid cancers are more common, okay, in women. Even so, males die at much higher rates and at much younger ages than females generally, due to all medical conditions. So there's gender equality, sex equality as far as the mortality statistics are concerned here. Okay, so well, if you're suspecting someone of having thyroid problems, let's say hypothyroidism, you will give them radioactive iodine to drink. Now, iodine is an element, and it's added to our salts. The common salt that you eat has iodine added to it, right? So um, this radioactive iodine is concentrated in your thyroid because the thyroid gland needs iodine to make its hormones. So it takes in all of the iodine. So your thyroid starts to glow, right? Now, if you find some areas in your thyroid which are not glowing as brightly as the others, what does that tell you? Not much hormone is being secreted by those areas. They're not as metabolically active, hypothyroidism. Or on the other hand, you might see some areas which are glowing bright red, a lot of color there. Those are too metabolically active. So that might be hyperthyroidism, even thyroid cancer. Because remember, cancers are more metabolically active. They steal oxygen and nutrients from their neighboring cells. So they glow brighter too. So you can use these radioactive isotopes for determining things like hypothyroidism or hyperthyroidism or cancers. Okay. All right. So here we are looking at uh, what I was mentioning before, the octet rule, right? What did I say? Well, atoms like to have 8 electrons in their outermost shells, okay? And not all atoms naturally have 8 electrons in their outermost shells. So how do they satisfy the octet rule? how do they make the number of electrons in the outermost shell 8? So we will take a look at how that happens. So let's try answer these questions first. What subatomic particles determine the mass of an atom? Protons and neutrons. Electrons basically have not much of a mass. What subatomic particles determine the charge? Now, protons and electrons. Protons are positively charged, electrons are negatively charged. Neutrons do not have a charge. Okay, so the atomic number is 17, which means 17 protons. Mass number is 35. So 35 minus 17, okay? Whatever that number is, that is the number of neutrons. We talked about that. Do isotopes represent the same element? Yes, they do. Do they have the same number of protons, neutrons, or electrons? They have the same number of protons and electrons, but a different number of neutrons. Describe a radioisotope. Radioisotope is a unstable isotope which is shooting out radioactive neutrons like alpha particles, beta particles, gamma rays, and breaking down into a more stable radioisotope. What is the relationship of the octet rule and chemical stability? All atoms like to have 8 electrons in their outermost shell. Once they have 8 electrons in their outermost shell, then they're stable. Till that time, they're still looking for those 8 electrons, okay? All right, so here we are looking at some other definitions, like what is called ions, okay? What are ions? These are charged atoms, either positively charged or negatively charged, all right? Also called electrolytes. So electrolytes that you would drink, like Gatorade or something for electrolytes, right? Like sodium or potassium or calcium, because they have a charge on them, right? Either positive or negative. And so what ions do is they maintain the electrical charge in the body, and they also are able to conduct electricity, and that's why they're called ions, okay? Because they either lose or gain electrons, okay? So if they lose electrons, they become positive because electrons are negative, right? If they gain electrons, then they become negative again because electrons are negative. So something to remember. Potassium ion, it's a positively charged ion. Positively charged ions are called cations. Negatively charged ions are called anions, okay? So potassium is a cation. It has a positive charge, and it's used for maintaining our electrical charge at a normal level. It's also needed for your heart to beat at its regular rhythm, okay? If you inject too much potassium into someone, the heart will stop, cardiac arrest. And that's what happens in cases of a lethal injection, right? So when we administer a lethal injection, it contains a high amounts of potassium chloride. And so this potassium, it detaches from the chloride and it affects your heart. So you stop, you die because of cardiac arrest there, all right? Okay, so not sure why. Okay, it's kind of annoying why it's showing like that, but okay, so what we have to remember here is cations are positively charged. And how I remember cations are positively charged is because if you look at the word cation, there's a T there, right? T. And if you look at the letter T, it kind of looks like a plus sign. So cations are positively charged, right? On the other hand, anions are negatively charged. And if you look at the first two letters here, N, like in the letter N, so N for negative. So anions are negatively charged, okay? So that's what we have to remember. Cations are positively charged because they have lost electrons. Anions are negatively charged because they gain electrons. That's something you have to remember. Okay, and these are examples of what forms cations and anions, as you can see. Okay. All right, so here we are looking at our first example of what is called an ionic bond. At the top, you can see that, right? Also called electrovalent bond. And the perfect example is given there using sodium and chlorine atoms, okay? So let's see how sodium and chlorine come together to form an ionic or electrovalent bond. Okay, so look at the sodium atom first. Let's look at the outermost shell. How many electrons does sodium have in the outermost shell? Just one, a single atom, right? How much would it like? How many would it like to have? 8. So it's missing 7, right? Okay. Now let's take a look at the chlorine atom here. Let's count the number of electrons in the outermost shell here. 1, 2, 3, 4, 5, 6, and 7. 7 electrons. How many would it like to have in order to satisfy the octet rule? 8. It's missing 1. So what do these two atoms do when they come in close proximity to each other. A perfect uh, type of relationship in exchange happens here. The sodium simply donates its single electron to the outermost shell of the chlorine atom. Okay. So by doing that, how many do we have here now for chlorine? 1, 2, 3, 4, 5, 6, 7, 8. So the chlorine has become chloride now, and it has become negative because again, it gained an electron, negative, so it's an anion. So it becomes a chloride ion now. What happened to the sodium? It let go of the only sodium, the electron that it had in its outermost shell. So the, now what we have is only this shell remaining, and this shell becomes the outermost shell now. And how many do we have in that shell? 1, 2, 3, 4, 5, 6, 7, 8. 8. So both of them are equally happy. They both got their desired 8 number. This satisfied the octet rule. Now, by losing that electron, the sodium atom became a cation, positively charged cation. Why? Because it lost an electron, right? So here is a positively charged cation, and here is a negatively charged anion. What happens when opposites come close to each other? They stick together. Opposites attract, right? Positive, negative. So sodium chloride, and that's a lasting union that happens between those two ions. All right, and this is an example of an ionic bond or an electrovalent bond. So here you see a number of sodium chlorides coming together, forming like a lattice crystal. And what are you looking at? What is sodium chloride? Table salt, common salt that we consume. And the average American eats way more table salt than they should, about 50 times more. So, and extra salt in your diet leads to hypertension, high blood pressure, right? People who are susceptible or sensitive to extra salt, they basically develop uh, hypertension or high blood pressure, right? So be mindful of how much salt you're consuming in your daily diet, not only in the foods that you cook, but also in processed foods like canned foods. There's massive amounts of sodium. So I personally, whenever I pick up any canned food or processed food off the shelf, I always check for the sodium content. And if it's some outrageous number like 500 milligrams or 600 or even higher, I basically avoid that, right? Uh, because extra salt leads to hypertension, eventually congestive heart failure. The heart starts to fail. And when that happens, they put you on a DASH diet, DASH. And that DASH diet, DASH stands for dietary approach to stop hypertension. All right. In other words, stopping high blood pressure or hypertension uh, using your diet. So salt-free foods, more fresh fruits and vegetables, beans, nuts, those kinds of things. All right. Stay away from red meat. Stay away from processed canned foods. Those kinds of things. All right. So list the common cations: sodium, potassium, calcium, okay, magnesium. These are all common cations. And anions: chloride, uh, phosphate. All right. These are all examples of common anions that the human body has. On the periodic table, highlight the element that form the common ions of the human body. So we can go back and do that. Explain how and why ions form based on the octet rule. We just went over that example. Could an ionic bond form between two cations? No. You have to have one positive, one cation, and one anion to attract each other, right? Okay. So next up, we have something called the covalent bond, okay? And now, even if you look at the term itself, covalent, what is it telling you? The outermost shells or the valence shells of two atoms are sharing, covalent, right? They're sharing their electrons together. So basically, that's what our next type of chemical bond is about, right? Um, electron sharing. So again, if you remember the first one, the ionic slash the electrovalent bond, what you did there was electron donation and electron acceptance. Sodium donated the electron, chloride or chlorine accepted it, and therefore they formed an ionic bond. Here we have an actual sharing. So how does that work? Let's go ahead and take a look. Um, Here in a minute. Before we do that, here we are looking at yet another term called isomers. So we talked about isotopes, right? Same element, same number of protons, but different number of neutrons. So what are isomers? Iso again means same, okay? Isomers are those substances that have the same chemical formula, but different structures or different structural formula, okay? An example, these three types of basic simple sugars, glucose, galactose, and fructose. Glucose is your blood sugar, galactose is milk sugar, fructose is fruit sugar, also found in honey, okay? So if you look at the chemical formula of all three of these monosaccharides or simple sugars, it's the same. Look, C6H12O6, 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 6 carbons, 12 hydrogens, 6 oxygens, no change. But if you look at the shape, glucose is has a slightly different arrangement. Look, here the H is at the top, OH, hydroxyl is at the bottom. Here the hydroxyl is at the top, H is at the bottom, right? So there is a slight change, as you can see. And then fructose has a different shape as well. So that's what isomers are. Same chemical formula, like that there, but different structures, a different structural formula, okay? All right, so let's talk about the covalent bonds now, all right? Here are 3 examples of what covalent bonds are all about. So if you look at the first one, it's showing you a bond between hydrogen, 2 atoms of hydrogen gas. Okay, so how many electrons does a hydrogen atom have in its outermost or valence shell? Only a single electron, right? So the only thing that they can share is a single electron. So they get 2 here, right? 2 electrons, and that forms a single hydrogen bond. Okay, that's how it works as far as a single covalent bond is concerned. Now, then you have something called a double covalent bond. Okay, and how does a double covalent bond work here? So if you have to look at these 2 oxygen atoms, each of these oxygen atoms has how many? 1, 2, 3, 4, 5, 6. 6 valence electrons. So how many do they need to make it 8? 2 more. What do they do? 2 oxygens come together and they share a double pair. Double pair these 4 electrons amongst each other in their outermost shell. So now count 1, 2, 3, 4, 5, 6, 7, 8. And the same for the other one. 1, 2, 3, 4, 5, 6, 7, 8. They're both sharing him. So that is a double bond because 2 pairs of electrons are being shared. So O2, all right, double covalent bond. All right, you also have a triple covalent bond, such as in nitrogen. Each nitrogen atom has 5 electrons in the outermost shell. So 1, 2, 3, 4, 5, 1, 2, 3, 4, 5 for each. So they each need 3 more, right? So they come together and they share now 3 pairs of electrons. Let's count now. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Octet rule satisfied for both. This is a long-lasting bond. This is an example of a triple covalent bond. As you can see here, triple, 3 lines, double, 2 lines, and then a single bond, a single line, as you can see here, okay? All right. So remember that 95% of bonds in the human body are of this type. They're covalent, and covalent is the strongest of the different types of bonds, okay? For carbon, each carbon has 4 electrons in its outermost shell, okay? 4 electrons in the outermost shell, so they need 4 more. And so they can get these 4 more electrons in a number of ways to form a covalent bond, such as look at this carbon. It has 4 in the outermost shell, so it can bond with 4 different hydrogens. Hydrogens have just 1, so 1, 2, 3, 4, 5, 6, 7, 8. 8 for carbon, and hydrogen, each hydrogen gets its 2 in the outermost shell. What you're looking at here is CH4 now. CH4, 1 carbon, 4 hydrogens. That is methane, which is kind of like the sewage gas or landfill gas. Methane in swamps and wetlands, because of bacterial action, methane is the gas that is released. Here, you're looking at a carbon dioxide molecule. How does that work? Carbon has 4 electrons in the outermost shell. Oxygens have 5. So 2 of those oxygens come and share their outermost shell electrons with the carbon. Now let's count. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Each one gets their desired number of electrons, as you can see here. Okay. All right. So that forms carbon dioxide, 2 oxygens. And then same here. Here, these two carbons are sharing uh, electrons with a number of hydrogen atoms, and then this oxygen is sharing electrons with both carbon here and hydrogen. What you see in common is each one is getting 8 electrons in the outermost shell. C2H5OH is the formula here, and this is a molecule of ethanol, which is a type of alcohol. So what we see is that carbon can form so many different shapes, and that's why carbon is the backbone of life, okay? Carbon is the backbone of life on this planet as we know it. And why is that? Because carbon is so versatile. It can change its shape. It can bond with so many other atoms. It's very flexible, like life itself. Life itself has to be flexible. If life is not flexible, then it goes extinct, right? Only living things that adapt to their surroundings are able to go on. So carbon is the backbone of life. And we just saw why that is the case. Based on its chemical structure of the atom, it can form its bonds in so many different ways. Look at that, carbon's a squiggly line molecule and a chain molecule here, right? Even rings, as you can see. So a number of different configurations that carbon can basically form. All right, so here we are looking at something called polar versus non-polar bonds, okay? And so what do we mean by polar bonds versus non-polar bonds? Let's take a look uh, at the water molecule, okay? And would have been easier if I could find a picture here. Okay, so in a water molecule, let me stop sharing for a moment here. Maybe I can explain it here. Okay, so here's water, right? You have what? You have H, hydrogen, right? And you have two oxygens here, okay? I'm sorry, and one oxygen, and then you have two hydrogens here. My mistake there. Okay, so now uh, what happens is oxygen is a much larger atom as compared to hydrogen. It has more atoms, 6 atoms in its outermost shell anyway, right? And hydrogens have only one. So since oxygen is a bigger, more powerful, stronger atom than hydrogen, what it does is it tends to attract more electrons towards itself, right? More electrons. It's kind of like a tug of war between the hydrogens and the oxygen. But since oxygen is bigger, it kind of wins, so it pulls more electrons towards itself and it becomes slightly negative, okay? So this is a delta negative sign, which means it's slightly more negative because it's able to pull more electrons towards itself. The hydrogens, they have to let go of the electrons, right? So they are not as strong. Therefore, since the electrons are pulled more towards the oxygen, each of the hydrogen becomes slightly positive because they're losing the electrons to the oxygen atom. So slightly negative oxygen, slightly positive hydrogens. Water is a polar molecule. Why is it called polar? Because it has two poles. That's what it means, polar molecule, right? Polar molecule, because there are two poles, a slightly negative pole and two slightly positive poles. These are, so this is what polarity is. This is a polar molecule, right? Uh, an unequal distribution of electrons here. Now, what else goes with the polar molecule? Oxygen is slightly more electronegative, and the hydrogens are more electropositive. That's what we have to remember, right? Okay, so then let's go ahead and share screen again. So here we are, right? And okay, now as an example, look at the not this, but this molecule here, fatty acids, right? So these are polar molecules. You can see water, right? Slightly negative, slightly positive polar molecules. Same with glucose, slightly positive and slightly negative poles. And so this is again a polar molecule. But if you look fatty acids, look at fatty acids here, right? There is no uneven distribution of electrons. They're all equally negative or equally positive or whatever. So there are no negative or positive poles in these types of atoms or molecules, and they're called non-polar molecules, okay? So One thing that you have to remember is water dissolves polar molecules. Polar molecules dissolve easily in water. Non-polar molecules like fats do not dissolve in water. And we know this, fats and oils and water don't mix, right? Uh, and so this is why people who have had gallbladder surgery, cholecystectomy, if you have taken out their gallbladders, right, because they might have like stones in there, gallstones or gallbladder infection or something, they are on a fat restricted diet. Uh, because if they eat a lot of fat, what does the gallbladder do? It stores a green bitter fluid called bile. Bile is produced by your liver, right? So when you don't have bile, because you don't have a gallbladder, it has been removed, and so you cannot digest and emulsify the fats in your diet that you're eating. So all of those fats pass out as is, without any absorption, into your stools. So these people who eat some fat after having their gallbladders removed, they get these fatty, smelly, greasy stools. And this is actually called, um, it's called uh, steatorrhea, okay? Steatorrhea. So let's say cholecystectomy, after cholecystectomy or cholecystectomy, which is gallbladder removal. Okay. Okay. So, so one thing that you will use to identify these fatty stools is, you see how they float when you go to the person goes and defecates. If you look at the toilet bowl, these first of all, they're very foul smelling, more foul smelling than usual, right? Because there's a lot of fat content in there, and fat tends to smell rancid. uh, but these stools also, they tend to float on top of the toilet bowl. They don't sink. Okay, why? Because fat is less dense than water, and fat and water and oil don't mix. Okay, so steatorrhea, again, same thing. Water only dissolves polar molecules, not non-polar molecules. So fats and oils do not dissolve in water. All right, there are some other types of bonds that we are looking at here, such as hydrogen bond. And hydrogen bond, again, can be seen here. Here you have water, right? So hydrogen bond is formed between the hydrogens, right? The hydrogens, the hydrogen atoms here, and the OH, the hydroxyl ions, the OH and the H's, the hydrogens and the hydroxyl ions is where you find hydrogen bonding. Okay, and this is a weak type of bond. It's nowhere near as strong um, as your covalent bond. Okay, what information about a molecule is gained by a structural formula? Uh, well, what type of a structure does it have? What atoms and in what configuration are those atoms arranged? How does a structural formula differ from a molecular formula? Molecular formula simply tells you the number of atoms, like C6, H12, O6, and glucose or fructose, right? Just the number of atoms, not how those atoms are arranged in the structure. What is an isomer? Uh, Two elements that have the same chemical formula, but different structural formulas. Explain covalent bond formation. It's all about getting 8 electrons in the outermost shell by sharing electrons. Once you get 8, or you satisfied the octet rule, that's when you have a stable bond there. Okay, assign the partial charges between nitrogen and hydrogen in a polar covalent bond. Hydrogen would be slightly positive, nitrogen would be slightly negative. Why are some covalent bonds non-polar and others are polar? Because in different bonds, sometimes you have slightly negative ends and slightly positive poles. So those are polar. In others, there's an equal distribution of the charges. So those are non-polar, okay? Are oxygen and carbon dioxide non-polar or polar molecules? They are non-polar because there's an equal distribution of charges. Okay. What is the name of the intermolecular attraction between a partially charged hydrogen of one polar molecule and partially negative atom of another polar molecule? Hydrogen bond. That's what they're called. All right, so next we are looking at the uh, what is called the universal solvent, or water, right? It's called the universal solvent because water dissolves most of the things uh, easily, the polar molecules at any rate, right? So as we can see here, water is the uh, basis of all life as well, right? Like carbon is the backbone of life. Water is the medium in which all life exists, as we can see here. So 2/3 of the human body is nothing but water by weight. If you have more muscle mass uh, than fat or adipose tissue, then you retain more water. If you have more fat or more adipose tissue, you tend to get dehydrated faster. So another incentive for losing some fat weight, right? The more muscle that you build, the more water that you can retain in the system. Okay, water is also the most important inorganic compound for living things. So at this point, let's talk about what is organic versus inorganic. So when I ask what is organic, the answer that I guess most commonly is, well, organic is natural or pure or something that exists in nature, right? And technically they're correct, right? But the scientific definition of an organic substance is if it is derived from a living thing and if it contains carbon, it's organic, right? So what is derived from living things? Chlorophyll, the green pigment from plants, that's organic. Sugar, glucose, it's made by plants, it's organic. Sweat, tears, saliva, blood, right? Body secretions, urine, semen, feces, whatever. These are all organic substances as well. So when you say organic, in biological terms, you're referring to something which is derived from a living source. Vitamins, they also are synthesized by living things. Bacteria make vitamins, um, other organisms make vitamins as well. So what is inorganic, and it contains carbon, right? So then what is inorganic, right? So let's quickly put it down here. So we're talking uh, organic versus inorganic. So for organic foods as well, if it's like natural, so-called, right, derived from a living source, not no chemical preservatives or additives, that's what it means. Not no artificial chemicals and preservatives. That would be inorganic. Organic is purely animal, like living thing derived, no pollution, and also containing carbon. Organic foods go bad faster, right? You have to eat it fresh, and they're good for your health because you're not ingesting all of those chemicals and pesticides and added preservatives in there, right? Uh, which is a risk factor for cancer as well, eating those kinds of preservatives. So, um, so organic stuff also smells bad to us. So if you think of any of these body secretions that I've just mentioned, or eggs or meat, they tend to smell bad after a while. That tells you it's organic. If it stays fresh forever and it has a very shiny, glossy look to it, such as some vegetables and fruits that you see in many of the outlets now, right? Um, and they don't go bad, it's not organic. They've added chemical stuff to it to increase the shelf life, not necessarily good for health. So don't compromise your health there, all right? Okay, so inorganic substances do not contain carbon by definition, with a few exceptions, and are not derived from living things like water, H2O. No carbon there, it's not derived from living things. Living things need water, water doesn't need living things. So that's inorganic, all right? Okay, so what do we know about water? It's a polar molecule. We talked about how oxygen is slightly more electronegative than the hydrogen there, right? It can form hydrogen bonds with adjacent molecules, as you can see here, hydrogen bonding in water, right? Here is a polar molecule. Oxygen tends to pull the electrons to a little too much towards itself, so it becomes electronegative, all of that. So water exists in three different forms, right? Solid, liquid, and gas. Solid water is ice, snow. Liquid water is water as we know it, and gaseous water is water vapor or steam, okay? And all three phases are imperative for life. How's that? So how is we know liquid water, we need to drink it because water is where all of our enzymes work. Water is a medium. It also regulates our body temperature. It gives us cushioning and friction resistance, right? It lubricates us and all of that. How about solid water? How does ice help life survive? Look at ponds and streams and lakes, right? So during the winter months, when it gets extremely cold, these large bodies of water tend to freeze up. So what happens when water turns to ice? This ice floats to the top. And this is a unique property of water. Generally, solid things are more dense than liquid things and they sink. But water is an exception. When water freezes over, it actually becomes less dense than liquid water. So ice floats to the top. So if you go to these rivers or lakes or streams and during winter months, they're frozen over and the layer of ice is at the top. But if you break through the ice, it's all liquid water underneath at a comfortable temperature. So fish in amphibians and all kinds of aquatic life can still survive because the ice rises to the top. It's still liquid water down there at a comfortable temperature. So that's another property of water that sustains life, okay? Um, and some animals can actually come back from the dead, if you will. Like there's a toad, it literally freezes over, right? There's a toad that, uh, let's call him the freezing toad, right? Uh, that literally freezes over. Uh, and what happens is, even though it freezes externally, internally it has an enzyme, which is kind of, it acts like a anti-frost, right? So let's call it the anti-frost enzyme. So this enzyme prevents uh, the insides from frosting up, right? So when the weather changes and it starts to warm up, the ice literally melts away from the toad, and its metabolism picks up again. So you have these kinds of examples in nature as well, right? So the freezing toad with the anti-frost enzyme inside of it, quite cool. So, and then moving on to the water vapor or the gaseous state of water, how can that sustain life? Well, if you look at the clouds, right? Clouds are a major part of the water cycle, right? And so have water clouds, that's gaseous water. Water evaporates and then it forms this layer of water vapor called clouds, and then it rains down again. So which is extremely important for replenishing the water cycle, right? Also, there's steam inside of your body, which keeps the insides of your lungs moist so you can exchange gases easily, okay? So gaseous water is needed for life as well. So here are some of the functions of liquid water. Um, as you can see here, all right. Okay, let's, right. So the other example that came to mind was that of something called the ice fish, okay. Uh, and these are species of fish that live in extremely cold waters, sub-zero temperatures, uh, like in the Scandinavian regions, right, the Northern Ocean and those kinds of places. So the ice fish are called that because if you look at their blood, it's completely clear, right? It's not red. Other regular fish have red blood like us, like other animals, right? But ice fish have literally clear blood, okay? And so what has happened in the course of evolution is that since hemoglobin is because of a protein, right? Red blood cells contain a protein called hemoglobin, and that protein in freezing temperature tends to clump up and form blood clots. So the ice fish have evolved to get rid of that hemoglobin for good, and they can directly absorb oxygen through their gills um, to the rest of the body. So they have disposed of the need for hemoglobin because that would form blood clots in them. Okay, so due to a genetic mutation, sometimes back in evolution, they developed this white, clear blood, and so now you have ice fish. So remarkable examples of how living things have adapted to the conditions where they live. All right, okay. So what does the water do? It transports stuff like blood and plasma, carries red blood cells, white blood cells, it carries even temperature, it carries oxygen, carbon dioxide, hormones, waste, all kinds of things. Water lubricates, like your joints have synovial fluid, your brain and spinal cord are protected by the cerebrospinal fluid, the peritoneal fluid in your peritoneum, all of that. It cushions, like a developing baby, an embryo or a fetus, which is surrounded by the amniotic fluid in the amniotic sac. It cushions the baby. It excretes wastes like urine. Urine is, of course, water that gets rid of waste, liquid waste. All right, so some properties of water that make it perfect for sustaining life. Cohesion, surface tension, and adhesion is what we are looking at here, okay? Let's talk about each. What is cohesion? Attraction uh, between water molecules for each other. And you see it all the time. For example, when you take a sip uh, through a straw, water rises up in a column. Why? Because one drop of water sticks to the other one and they follow each other in line, right? So you can sip. If there's an air bubble in your straw, then that column is broken and the water is not going to rise anymore. You have to clear out the air bubble so that the water can resume flowing, all right? So cohesion, water molecules stick together. You can see it. So how does it sustain life? Your blood flows in a line through your arteries and veins, right? Because the water molecules in your blood are sticking together just like drinking water through a straw, they're going in a straight line. If you get an embolism, which is a clot, an embolism is a clot. It could be a blood clot, it could be a cholesterol like a fatty plaque, it could even be an air embolism. If someone injects you with an empty needle with air, those air bubbles will break your blood flow. So once that blood flow is broken, now you don't get blood supply to your body, you can get a stroke or a heart attack or some kind of embolism, pulmonary embolism. If this forms in your lungs, you die because you cannot breathe properly due to a pulmonary embolism. Even fat embolism can happen. For example, if you fracture one of your major bones, fat could leak from there and form fat emboli. So let's uh, talk about types of emboli, which are clots, right? So we talked about uh, fat embolism, we talked about air embolism, right? Uh, fluid, like amniotic fluid embolisms. So that's what happens. So you need to maintain this steady stream of blood supply in your blood vessels. That's cohesion. Same for trees. Trees suck up water through their roots, just like using a straw. So it's a constant stream of water because they stick to each other. All right. Okay, so then we have adhesion. What is adhesion? It's the attraction of water molecules with some other substance. Some common examples: ever tried pouring water out of a glass or a container into a different glass? What happens with the final few drops? You really have to shake the container or the glass to get those to shake free and then go to the other glass, right? Why? Because those drops of water are sticking to the glass, right? So you really have to shake it. So that's adhesion. How does that help human life? Well, your blood, which is made up of 55% of water, plasma, right? Plasma is 92% water. That tends to stick to the walls of your blood vessels. So it keeps flowing by attaching to the walls of your blood vessels. If there were no adhesion, water will fall loose and your blood vessels won't be able to pump blood across your body. So there. Then surface tension. What is surface tension? Surface tension is the ability of water to kind of form a sheet because of cohesion. Again, water molecules attract each other, so they kind of form a sheet, okay? One perfect example of surface tension is when you take a glass of water, and if you release a pin or a needle, don't drop it, but just release it gently, it'll keep floating at the top. It's almost like a sheet is holding it up. As soon as you touch it gently, you break the surface tension, the needle is going to drop, sink to the bottom. That's surface tension. Another dramatic example of surface tension is I don't know if you have ever heard of the Jesus lizard, right? The Jesus lizard, which is the name given uh, to a species of South American lizard called the basilisk. And the Jesus lizard is called that because it can run across water. At high speeds, it can sprint across like ponds and lakes in pursuit of its prey or escaping from danger or something, or from a predator or something like that, right? So it's like a miracle of walking on water. How does it do that? Well, part of the answer is because it has these large feet, and there's like air bubbles form under the feet, and it runs at such top speeds that it literally skims over water. The air bubbles keep it on top, but the part of the answer lies within the property of water itself. Water tends to form a sheet that holds the lizard up, right? Um, water skippers, water striders. These are insects that do the same. You might have seen them in your swimming pools or somewhere. These insects that literally walk on water, the water striders, right? And so they again uh, are making use of the property of water called what? Called surface tension. Okay, water striders are interesting creatures. The male water strider, when it times come for mating, the male water strider will go to the female, and if the female rejects the advances of the male, it has a blackmail trick up its sleeve. What it does is it starts tapping its leg on the water body gently, which creates ripples and vibrations, which attracts predatory fish. So in other words, it's blackmailing the female. If she doesn't allow him to mate with her, it's going to tap the water, attract the predatory fish, and then escape. And the female is going to be gobbled up and devoured by the fish. So yeah, it exists in nature as well. An actual example of blackmail, a sexual blackmail, or whatever you want to call that, right? Very interesting. So let's take some notes here as well. Okay, so how does that translate into human anatomy and physiology? Surface tension. Okay, now your lungs. Our lungs are wet on the inside. They're not dry. We need moisture for oxygen and carbon dioxide to be diffused across the lungs. You cannot have dry lungs, okay? But the lungs being wet on the inside also poses a problem. If you've ever tried blowing up a dry balloon versus a balloon which is wet on the inside, which one is easier to blow up? The dry balloon, right? You can blow it up much easily as compared to the one which is wet, because wet balloon tends to stick together. So you really have to blow hard in order to inflate it. So same with the lungs. Since the lungs are wet on the inside, they tend to stick together to collapse. But with each breath, you want to open your lungs up, right? So it's quite a challenge to open up lungs which are wet on the inside. So how do you get rid of this? Otherwise, you'll have to snort and a lot of effort breathing every time in, right? So how do you get rid of this? Fortunately, there are some cells called the alveolar cells inside of our lungs, which secrete a fatty substance called surfactant. Surfactant. And what this fatty substance, oily, viscous substance called surfactant does is it prevents your lungs from sticking together, from collapsing. This fatty substance, again, fat and water don't mix. So it prevents your lungs from sticking together so you can breathe easily. Now, in children that are born premature, preemies, premature born children, they are predisposed to a condition called IRDS, which stands for infant respiratory distress syndrome. Okay, preemies, prematurely born children are predisposed to this. So if you are working in the neonatal ICU or something, right, or in the delivery room as an OBGYN, and you see this preemie, which there's flaring of the nostrils, and he or she's turning blue in the face and not responding and having a hard time breathing, consider IRDS, infant respiratory distress syndrome. So what happens in these kids is, since they're born in such an immature state, their lungs do not open up properly. The lung cells do not produce enough surfactant, so the lungs collapse because of wetness, and so they can't breathe properly. Fortunately, there's an easy fix. We have synthetic, man-made, artificial surfactant, which can be introduced into the lungs, and then the lungs start opening up, and they can breathe as they should. All right, some other properties of water. Water has a very low specific heat. So what is specific heat? As you can see here, it's the amount of energy required to increase the temperature of 1 gram of substance by 1 degree Celsius. For water, the specific heat is uh, actually quite high, not low, but quite high, which means you need a whole lot of energy, whole lot of temperature of heat to increase the temperature of 1 gram of water by 1 degree Celsius, okay? So how is this a good thing for life? Well, since water has such a high specific heat, you can heat up a whole lot, and just a few drops of sweat will take all of that heat away from your body, right? Because water has such high specific heat, you need a whole lot of temperature and heat to increase the temperature of just 1 gram of water, okay? So high specific heat keeps you from being dehydrated. Just a few drops of sweat will rid you of massive amounts of heat. So you're conserving water in the end. Same with heat of vaporization. Water has a very high heat of vaporization. What is heat of vaporization? It's the heat required for release of molecules from a liquid phase into a gaseous phase for 1 gram of a substance. Again, since water has a very high heat of vaporization, a few drops of sweat will cool off by releasing massive amounts of heat from your body. And therefore you end up uh, conserving water, okay? You don't dehydrate. Water is called the universal solvent because most of the substances will dissolve in it. Only non-polar substances like fats and oils do not dissolve in it, okay? All right. So water substances that are attracted to water or dissolve in water are called hydrophilic. Philia means love, so hydrophilic substances dissolve in water. Hydrophobic, phobia means a fear. So hydrophobic substances do not dissolve in water. They don't like water, okay? So for example, fats and cholesterol, as it says here, those are hydrophobic, so they don't dissolve in water, okay? But then you also have substances which are amphipathic, which means they are part polar and part non-polar. Parts of them dissolve in water, the other parts don't, repel water. And a perfect example of an amphipathic molecule is your cell membrane here. Cell membrane is made up of phospholipids. The phospho part is hydrophilic. It likes water, so it faces towards water, like these two purple ends, right? And the lipid part, which is in the middle, lipid is fat. Fat doesn't like water, so it's facing away from water here in the middle. See how the phosphoheads are facing towards water, and the lipid tails are centered in the middle. So your cell membrane is an amphipathic molecule, part water loving, part water hating. So what is the intermolecular bond that is significant in determining the properties of water? Hydrogen bond. Which property of water contributes to the need to produce surfactant and prevent collapse of the alveoli in your lungs? Surface tension. Which property contributes to body temperature regulation through sweating? High heat of vaporization. Why is sweating less effective in cooling the body on a humid day? Because there's already so much water in the air, not much sweat can be lost. So if you live in Florida or someplace, it's quite humid there. So you tend to get sticky. But if you're in a dry place like Central Texas or North Texas, then you dry out completely because the air is dry. How does the interaction of a non-electrolyte in water differ from the interaction of an electrolyte in water? Electrolytes tend to break down into positive and negative charges, cations and anions, and they can conduct electricity. Non-electrolytes like sugar, they can dissolve sometimes, but they do not break down into ions, cations and anions, so they don't conduct electricity. How do phospholipid molecules interact with water to form a membrane? Phospholipid molecules are amphipathic. The phospho part likes water, so faces water. The lipid part dislikes water, so faces away from water. All right, so next up, we are looking at, and it's again doing this funky number. Okay. We are looking at acids and bases, okay? So what is the difference? What are acids and bases, and what is their function in maintaining life, okay? So let's look at acids first. First up, acids are substances that generally taste sour, though you should not be tasting acids because some of them are quite corrosive. They taste sour. When dissolved in water, they release hydrogen ions, H positive ions. That's one property, okay? And acids uh, tend to turn litmus paper from blue to red color. Litmus paper are these colored paper strips. So acids turn them from blue to red, okay? So how I remember what acids do is, so let's say acids, they release hydrogen ions, right, in water. They turn litmus paper blue to red. And how I remember that is by using the mnemonic bracid, right? Bracid means blue to red for BR, bracid. Acids turn litmus paper blue to red. So remember that. They also generally taste sour, right? And what else? They are proton donors. They release protons. That's another property of acids, as we see there. And they also have a value of less than 7 on the pH scale. So acids give you a value of less than 7 on the pH scale. Okay, so these are some properties of acids. Where do you have acid in the body? In your stomach. Stomach acid, that's hydrochloric acid, which helps you with digestion. The vagina, the female birth canal is also acidic in pH. And that acidic pH in the female birth canal prevents infections. During menstruation, during childbirth, during sexual intercourse, all of those things, the normally acidic vaginal pH prevents microorganisms, germs from growing there, okay? So now let's take a look at bases. Bases are the exact opposite. So first step, bases release not hydrogen ions, but OH negative or hydroxyl ions in water, number one, okay? Bases turn litmus paper the opposite color. Turn litmus paper from red, turn litmus paper from red to blue, okay? What else? Bases generally taste bitter, unlike acids that taste sour, right? Bases also are proton acceptors. Rather than proton donors, they accept electrons. And then finally, bases give you a value greater than 7 on the pH scale. Okay, that makes a base. So those are the differences between acids and bases. Okay. So what is the pH scale? Let's go ahead and take a look. Here's the pH scale. All right, a pH scale determines if a substance is acidic or basic. So the pH scale goes from 0 to 14. What is the midpoint? 7. right? So any substance that falls on 7 or around 7 is neutral. It's neither acidic nor basic. The lower down that you go from 7, 6, 5, 3, 4, 3, 2, 1, so on, the more acidic. The greater than 7 that you go, 8, 9, 10, 11, and onwards, the more basic the substance is. So look at the most acidic substance in existence, hydrochloric acid, which has a pH of about 1. That's your stomach acid, okay? Uh, so how come such corrosive acid is not burning holes through your stomach? Sometimes it does when you develop peptic ulcers or gastric ulcers, but you have this thick coating of alkaline mucus that neutralizes this acid and protects the walls of your stomach from being dissolved by the strong acid, okay? What is the strongest base in existence? NaOH, sodium hydroxide, which gets a 14, the maximum number on basic substances or alkaline substances, all right? So this is the pH scale, basically, that tells you whether something is neutral, acidic, or basic. Water is classically neutral. Water is here. The blood is also anywhere from 7.35 to 7.45. So almost slightly alkaline, but almost neutral. So blood, saliva, as you can see here, right? Milk and saliva are slightly acidic. Pure water is 7. Human blood is slightly alkaline, 7.4, right? Seawater is about 8, so on and so forth. So, all right. Now, that also reminds me of one of the most gruesome cases that I saw during my medical ICU rotation. And this was a young gentleman in his early 20s who had tried to commit suicide by drinking a whole container of the plumbing solution called Drano, right? So let's make a note here. Drano suicide attempt in medical ICU, right? And so he had suffered some heartbreaks in his love life or whatever. So when people found him, he had ingested a whole container of that Drano. And that is sodium hydroxide. That is a very strong corrosive base. So what that had done is it had literally like dissolved and melted away the lips and the surrounding facial tissue of the patient. It literally had gummed up, right? And you could see the teeth exposed there because everything else had been just burned away. And even the esophagus was all burnt out. So how did we manage this patient? The first challenge was to keep the patient hydrated and fed, and also to maintain their pH balance, right? To prevent too much alkalinity here. So we started infusing IV fluids into him, all right? We started normal saline, 0.9% normal saline, which is a salt solution. We started pumping him with that. We also added, later on, an IV fluid called Ringer's lactate. Ringer's lactate is another type of IV fluid which also has nutrients like calcium, magnesium, fatty acids, amino acids, those kinds of things, all right? So it has electrolytes, sodium, potassium, calcium, okay? Amino acids, fatty acids. So that's an ideal solution for feeding someone intravenously. Obviously, you couldn't give him anything per orum, which means through the mouth, fatty acids, because obviously there was no mouth as such. And then we had to like schedule him for facial reconstructive surgery. And so they were able to reconstruct the facial structures and we were able to save him, okay? So one case that I was reminded of here. So what is neutralization as we see here? Acids and bases, they neutralize each other, right? So if you have strong acid in the body, you have substances called buffers, which prevent this fluctuation in the pH. They neutralize the pH, okay? They keep your body from getting too acidic or too basic, all right? That's what buffers are, and therefore they help maintain your body pH or your blood pH in between 7.35 to 7.45, as you can see. So explain why water is neutral, because when water has an H2O, when broken down, it has an equal amount of H positive hydrogen ions and OH negative and hydroxyl ions. So since H positive and OH negative are in equal numbers, it's neutral. Which type of substance releases hydrogen ions in the water? Acids. What is the general relationship of hydrogen and pH? The greater the number of hydrogen ions released by a substance, the more acidic it is, the lower the pH. Remember, acids are lower than 7. So the higher the hydrogen ions, the lower the pH. It's an inverse relationship. Why are buffers important? Because they prevent fluctuation of your pH. They prevent your body from getting too acidic or too basic. How do they function to help maintain pH? That's what they do. All right, so here we're looking at different types of mixtures like suspensions, colloids, solutions, uh, and emulsion. So what are they? Let's take a look. Using blood as an example. All right, so what is a suspension, first of all? A suspension is a mixture where some solid substance is suspended in a liquid substance. Perfect example, sand and water. If you take a bunch of sand, put it in a glass of water and shake it, stir it, it'll form a suspension because the uh, sand molecules, sand particles will be suspended in the water, right? And your blood can act like a suspension too, because in what is blood? Blood has your blood cells, white blood cells, red blood cells, and platelets, which are suspended in water. So blood itself is an example of a suspension as well, all right? So sand in water, suspension. Then what is a colloid, as you see here? The perfect example of a colloid, imagine you crack open an egg, put it in a glass of water and stir it. So the egg is kind of like a protein and it will form a colloid in that water, right? Same with gelatin. If you take gelatin and if you dissolve it in water, same thing. You come up with a colloid, all right? And egg, by the way, is superfood. Eggs are termed as superfoods, right? Because uh, earlier, there was some discussion of whether they're too high in cholesterol and whether is it safe to eat them on a regular basis or not. Now, the scientific medical consensus is that it is perfectly safe, in fact, healthy to consume for most people one egg on a daily basis, all right? Because eggs provide essential nutrients to you, including the three extremely important proteins, all right? What are those important proteins? Albumin, fibrinogen, and globulin, all right? Each one has critical functions in your body. Albumin is the most abundant protein in the human body. And what albumin does is it gives shape and tonicity to your cells, gives shape and tonicity to cells. It maintains your cell structure, your cell integrity. Albumin comes from eggs. It is found in the egg white. Albumin is egg white. Egg white is albumin. Then you have fibrinogen. Let's talk about globulins first. Globulins are again found in egg white, and globulins are an important part of your immune system, like your antibodies, right? Globulins are used in your immune system to form antibodies. So even if you look at the term for antibodies, they're also called immunoglobulins. Look at the term, immunoglobulins. They are made up of proteins called globulins, which you get from eggs, egg white. Finally, fibrinogen from egg. Fibrinogen is found in the egg yellow part or the egg yolk. And fibrinogen are important because they help your blood clot, clotting factor, okay? And your blood clotting is very important because you don't want to bleed from a cut, right? So that is fibrinogen, and that comes from eggs as well. If you've ever cracked open an egg, what happens to the yolk first? It's like kind of runny, but what happens to the yolk after a while? It clots. Have you noticed it? It clots up because of fibrinogen, and the same thing makes your blood clot too. So that's why eggs have been termed superfood. Okay, and then you have a solution. Your blood can act as a colloid as well because blood is nothing but egg white and all kinds of proteins suspended in water. So so your blood can act as a suspension. Your blood can also act as a colloid. So then what is a solution? Solution, perfect example, take salt, put it in a glass of water, stir, and you get a salt solution like 0.9% normal saline. That's a solution. And your blood is a solution too, because you have salts dissolved in water in your blood as well. And then finally, you have something called an emulsion. An emulsion is like if you put oil and water, they don't mix, but they kind of form this like murky, milky substance, right? Emulsion, oil and water. And that is again true for your blood as well. Your blood can be considered an emulsion too, because then you have these fats, these lipids, fatty substances, which are basically suspended in water. Okay, so when left standing, red blood cells settle to the bottom of a tube of blood. Based on the characteristics alone, blood would be characterized as a what? Suspension, colloid, or a solution. So here we are looking at the red blood cells which are settling to the bottom. This is a suspension, like sand and water, right? If you if you stir the sand in water, let it stand, the sand will gradually deposit to the bottom. So same with your red blood cells. Here, your blood is acting like a suspension. Why is blood also considered the other two types of water mixtures? Because colloid, because there's also protein dissolved in blood, and solution, because there's also salts which are dissolved in blood. So, all right, so here we are looking at what are called biological macromolecules or biomolecules. In other words, molecules that make up life, or life needs to sustain itself, right? And the thing to remember about these biomolecules is they have certain elements in common, right? So again, if you remember the acronym CHOMPS, okay, CHONPS, all right? So that will help you remember that the biomolecules all contain carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, all right? So again, the fundamental basis of life based on these elements, CHOMPS, remember that acronym here, as you can see, okay? And then hydrocarbons, again, carbon is common in all living structures. Hydrocarbons contain only carbon and hydrogen, as you can see here, okay? Most of these biomolecules are polar. They have a positive and a slightly negative pole, okay? Some are acidic, some are basic. So there's some variation there as well. Okay, so then polymers, what does that term mean? A polymer is a long chain molecule made up of smaller particles called monomers, okay? For example, all carbs, carbohydrates, which are a type of biomolecule, are made up of glucose monomers. Many glucoses come together to form a long chain uh, polymer, which could either be starch, if it's in plants, or glycogen. Glycogen is how animals store glucose or sugar. Um, Proteins are made up of these small, proteins are, again, polymers, long chain molecules made up of these smaller monomers called amino acids. Fats or lipids are made up of fatty acid monomers. Nucleic acids are made up of nucleotide monomers, as we see here, okay? So what we are looking at here is how you can break apart these long chain polymers into smaller pieces, or how you can make a long chain molecule from smaller pieces for that matter. So first up, we're looking at something called dehydration synthesis, also called condensation synthesis. Okay, so why is it called that and what is it? If you look, take a look here, two monomers come together, right? This monomer releases a hydrogen ion or a hydrogen atom. The other one releases a hydroxyl ion or OH. So H and OH, that gives you H2O. In other words, one molecule of water has been released, and as a result, these two monomers join together, forming a dimer, like two of them together. So this is dehydration or condensation synthesis. And you see why it's called that, because you're literally getting rid of water here, right? And joining these two together. The opposite of dehydration or condensation synthesis is something called hydrolysis. Now look at the word itself, hydrolysis. Lysis means to break apart, like Lysol literally breaks apart the bacterial cell membranes. Lysis, hydro is water. So in other words, adding water to a long chain molecule or a dimer with two particles to split it apart. This is the exact opposite of dehydration synthesis. You have added water here and you have split this dimer into its individual monomers. So these are two opposite reactions, dehydration or condensation synthesis and hydrolysis, as we see here. Okay. Using a different color than you did, highlight the 6 common elements that form CHOMPS, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. What functional groups may act as an acid? Those that release hydrogen ions act as an acid. Fatty acids, amino acids, right? What defines a polymer? Long chain molecule. List the 3 biological molecules that are macromolecules that are polymers: proteins, fats, carbohydrates, and the monomers that compose them. So carbohydrates are composed with glucose monomers, proteins are composed of amino acid monomers, and fats or lipids are composed of fatty acid monomers, okay? Let's start with lipids or fats first up. So lipids includes fats, waxes, and oils. What's the difference between fats and oils? No difference. Fats are solid at room temperature, oils are liquid at room temperature. That's the only difference. For example, if you take butter, a dab of butter out of the fridge, it's solid. But if you leave it on the table for a while at room temperature, it melts into an oil. That's it, all right? So there are 4 primary classes of lipids, as you can see here: triglycerides, phospholipids, steroids, and eicosanoids, all right? So what are triglycerides? Triglycerides is how you store extra fat, how you store extra calories. Anyone who's eating more calories than they're expending will have a high triglyceride level. I, for one, suffered from hypertriglyceridemia, all right, which is high triglyceride levels about 10 years back, right? And so again, what that told me was a couple of things. Number one, of course, I was eating more calories and not being as physically active and exercising to use those calories, right? So that was part of the answer. My diet, which was rich in calories and not enough exercise. But there was another equally important part that I found out later on. I had the genes for hypertriglyceridemia. It's also a genetic thing. So if you inherited the gene for high triglycerides, then that also contributes to the picture. So what's the, and hypertriglyceridemia is a cardiac risk factor. If you're too high of triglycerides, they're gonna clog up your blood vessels and you are at high risk for heart attacks and stuff, right? So the treatment is, again, diet and exercise. Be more physically active, eat smaller portions of food, more fruits and vegetables and nuts and beans, nuts, olive oil instead of like some canola oil and those kinds of things. Also be more physically active, lose weight, right? Not much you can do for the genetic part. Um, Fish liver oil is also shown to be effective. Fish liver oil is also shown to be effective in lowering your triglyceride levels. If that doesn't work, then medication like niacin and fibrates. These are two classes of drugs that can be taken to lower your triglyceride levels, okay? So then triglycerides or fatty acids can be saturated, unsaturated, or polyunsaturated. What's the difference? Saturated fats are solid at room temperature. They're animal fats, like what the stuff you have in steaks and hamburgers and all that. That's the dangerous type of fat, because the more you eat that, the more visceral fat or abdominal belly fat you're going to have, right? And that's a risk factor for diabetes, heart disease, strokes, cancer, metabolic syndrome, sleep apnea, you name it, and it's a risk factor. Unsaturated fats have a double bond at least, and they're usually liquid like oils, right? Saturated fats only have single bonds. They do not have double bonds. Polyunsaturated fats have two or more double bonds, as you can see here. All right, lipogenesis versus lipolysis. Lipogenesis is when you put on fat. Genesis, making of fat. Lipolysis is breaking down fat. When you lose fat weight, you are in lipolysis. When you accumulate fat in your body, you're in a state of lipogenesis, okay? So next up, we are looking at phospholipids here, all right? So the first type of lipid that we are looking at here are phospholipids. And as you remember, phospholipids are what make up our cell membranes, right? They're amphipathic, meaning they have a water-loving part, which is the phospho part, the phosphates, and then the water-hating or hydrophobic part, which is the lipids or the fatty tails, all right? So that is how our cell membranes are arranged. The other type of lipids that we are looking at here are called steroids. Steroids are fat-derived, or more appropriately, cholesterol-derived substances that have many different functions, like hormones in the human body, all right? So steroids um, come from cholesterol, and cholesterol is only derived from animal foods. Plants do not have cholesterol. Rather, they have something similar called sitosterol, but not cholesterol. So if you're a vegan, as an example, you would not be getting any cholesterol from your diet, which is just as well, because our livers make all the cholesterol that we need, okay? It derives it from even the plant sources that we eat. However, on top of that, we can eat moderate to small quantities of cholesterol. The problem is the average American eats far more cholesterol than what is considered little amounts or average. So the result being all of that extra cholesterol gunks up your arteries, basically leading to heart attacks and strokes and all those kinds of things, okay? So that's what steroids basically are. Some examples like testosterone and estrogen, the sex hormones, are also derived from cholesterol, right? They're examples of steroids. Eicosanoids is another category of lipids found in humans. These are modified 20 carbon fatty acids, as you can see here, okay? And there's many different types of eicosanoids, as you see here: prostaglandins, prostacyclins, thromboxanes, leukotrienes. Each one has different functions. Uh, Prostaglandins, for example, can cause your smooth muscle to contract. Okay. Prostacyclins and thromboxanes are involved with blood clotting. Leukotrienes, again, are involved with things like inflammation and so on. All right. Do lipid molecules typically dissolve in water? No, they don't. They're hydrophobic and non-polar. Which class of lipids form cell membranes? Phospholipids. What characteristic allows it to perform this function? It's amphipathic, meaning part of it is hydrophilic, the other part is hydrophobic. So we talked about this, how most animal fats are saturated. They have a single bond between carbon atoms and they are solid at room temperature. Vegetable fats, most of them are liquid uh, or unsaturated, having at least one double bond. They're generally healthier because they don't gunk up and clog your arteries as much, okay? But they can be converted to saturated fats by hydrogenation. And that can also lead to the generation of trans fats. Trans fats are some of the most harmful substances that we consume. They increase the risk of heart disease and stroke. So things like chips and cakes and pastries, those kinds of things tend to be high in trans fats. So something to be avoided. All right, next up we have carbohydrates, and they're called that because they have carbon, hydrogen, and oxygen, right? And the chemical formula always is CH2ON. So the carbon and oxygen have the same number of atoms, while the hydrogen has twice the number of atoms there. So some examples of carbohydrates, let's start with monosaccharides. Monosaccharides are simple sugars, okay? Glucose, that's a blood sugar. That's how we uh, regulate or the form in which sugar is accumulated in the body, right? It's a 6-carbon carbohydrate, most common monosaccharide as well. The primary source of energy for our metabolism is glucose. If you have too much of it, you end up with diabetes mellitus. If you have too little, you get hypoglycemia. So glycogen is how glucose is stored in the human and animal tissue, right? So many glucoses come together, the monomers, to form a long chain polymer called glycogen, and it's stored in your liver and in your skeletal muscles, okay? So again, the more muscle you have, the more sugar you can store, the lesser your chances of getting diabetic. So this glycogen can be broken down when needed, such as when you're starving, to release more glucose, and that's called glycogenolysis, as you see here, okay? Or uh, you can also get glucose from non-sugary foods as well, and that process is called gluconeogenesis. That happens in the liver. It can also happen in the kidneys. So when you're starving, glycogenolysis and gluconeogenesis are two substances which will provide energy needed there. All right, so what happened? Let's see there. Okay, all right, so then next here, single glucose molecule, C6H12O6, 6 carbons here. That's the monomer. You put many of those together, you get glycogen. Plants cannot store glucose as glycogen. They store it as starch. So starch only comes from plant foods. Okay, animals don't store that. Okay, so then there's different examples of these simple sugars like hexose monosaccharides. Hexose for 6, they have 6 carbon sugars like galactose and fructose. We also have 5 carbon pentoses, pent for 5, like ribose and deoxyribose, which are the base sugars in DNA and RNA. And disaccharides, where 2 monosaccharides are joined together by dehydration or condensation synthesis, such as sucrose. Sucrose is 1 glucose combined with a fructose, that's sucrose. Lactose is 1 glucose combined with a galactose. And maltose is 2 glucoses combined together. So these are disaccharides, okay? Polysaccharides are again long chain molecules of sugar. Glycogen is how you store sugar in animals and plants, in animals and humans. Starch and cellulose is how you store polysaccharide glucose in a long chain form in plants, okay? So cellulose, again, is a type of fiber, and we need to include fiber in our diet. Fiber you get from fruits, from vegetables, plant sources, and even though you don't digest fiber, but fiber makes you feel full. So it provides bulk to your intestine, so you don't eat that much. Fiber also takes away cholesterol. It coats cholesterol in it, and you defecate the cholesterol out. So it also removes cholesterol. Fiber is strongly protective against colon cancer, colorectal carcinoma, which is the 3rd most common and deadly cancer in men and women after lung and prostate. prostate and lung in males, and breast and lung cancer in females. Okay, something to remember. So here are some examples of monosaccharides: 6 carbon sugars like galactose and fructose, 5 carbon ones, ribose and deoxyribose, and disaccharides: sucrose, lactose, and maltose. So what is the repeating monomer of glycogen? Glucose. Where is glycogen stored in the body, in the liver, and skeletal muscles? For each of the following, indicate if it's a monosaccharide, disaccharide, or polysaccharide. Fructose, monosaccharide. Galactose, monosaccharide again. Glucose, monosaccharide. Glycogen is a polysaccharide. Lactose, two of them together, disaccharide. Maltose, disaccharide. Starch, polysaccharide. Sucrose, disaccharide. So then you have nucleic acids. They're called that because they're found within the nucleus of cells, right? There's two types of them, DNA and RNA for deoxyribonucleic acid for DNA and ribonucleic acid for RNA. Each of those are made up of, these are long chain molecules or polymers again, they're made up of simpler monomers called nucleotides, right? Each nucleotide has a 5 carbon sugar. If it's DNA, then it will be a deoxyribose sugar. If it's RNA, then this would be a ribose sugar combined to a phosphate group and also combined to a nitrogenous base. And these nitrogenous bases are based on 4 letters: A, G, T, and C. A for adenine, G for guanine, T for thymine, C for cytosine. All right. So that's basically what a single nucleotide looks like. And nucleotides are the monomers of nucleic acids. All right, so there you have those letters, right? 5 types of nitrogenous bases: A for adenine, G for guanine. These two are called purines, and these are double ring bases. Cytosine, thymine, and uracil. These are single ring bases, and these are called pyrimidines. All right, so 2 types of nitrogenous bases that you might come across. So here in this picture, you can see this is what cytosine, uracil, and thymine look like. See, they are single ring, and then guanine and adenine, these are double ring structures, as you can see. All right, so purines and pyrimidines. So DNA is the basis of life, right? DNA is how we pass on our genetic instructions to our offspring, all right? DNA is found in chromosomes, which are found in pairs inside of our cells, all right? It's also found in mitochondria inside of the cells. It's made up of these 4 letters, nitrogen bases, 5 carbon sugar called deoxyribose, and phosphate, all right? One thing to remember is DNA, in DNA strands, T always go with A with T, adenine with thymine, and cytosine always pairs with guanine, A with G. So one way you can remember is this is by memorizing the acronym apples on a tree, right? Apples on a tree. So A for apples, A goes with tree, T for thymine on a tree. So A, apples on a tree, A with T, and cars in a garage. So again, cars with a C, so cytosine goes with, garages with a G, so cytosine goes with guanine. All right, apples on a tree, cars in a garage. A bonds with T in DNA, and G forms with C. So RNA is similar. The only difference here is RNA is a single strand molecule. It's not a double stranded molecule like DNA. All right. And in RNA, you have ribose sugar, which is a 5 carbon pentose sugar, unlike deoxyribose in DNA, a phosphate, and one of these 4 nitrogenous bases: adenine, guanine, cytosine, and there's no T here, no thymine. Instead of thymine, you have something called uracil, U for a uracil, okay? So here's the difference between DNA and RNA. You see that DNA is a double-stranded structure. A is going with T always, and G goes with C. RNA, on the other hand, is a single-strand structure, and there is no T here. There's a U instead of T, okay? The sugar in DNA is deoxyribose. The 5-carbon sugar in RNA is ribose. So these are the differences in between these two genetic materials. Then you also have something called ATP, which stands for adenosine triphosphate. ATP is how we generate energy in our body to keep us alive for all of the metabolic functions to take place. It's by means of ATP. In an ATP molecule, adenosine triphosphate is made up of 1 molecule of adenosine, which itself is made up of 5-carbon sugar ribose, just like in RNA, and a nitrogenous base, which is adenine in this case, and then 3 phosphates. So therefore, adenosine triphosphate. Tri means 3. So 3 phosphates attached to this adenosine here, right? So you might be wondering, okay, so how do we get energy out of this molecule, right? You get this energy by detaching or getting rid of one phosphate. When you release or eject one phosphate from here, that releases a lot of energy. So, and then you're left with something called ADP for adenosine diphosphate. Di means two. So only two phosphates remain, and that phosphate is kicked off, and that's when you get energy. Okay. So ATP is our energy carrier. General function of nucleic acids to carry genetic instructions, inheritance. Structural differences between RNA and DNA. RNA is single-stranded, DNA is double-stranded. RNA has a uracil instead of a thymine or T. DNA has thymine instead of uracil. The sugar in RNA is ribose. The sugar in DNA is deoxyribose. Three differences. So next up, we have proteins. And proteins have different functions, as you can see here. Proteins are made up of monomers called amino acids. Proteins themselves are polymers, long chain molecules, and they serve a number of functions as we see here. They serve as catalysts. What is a catalyst? It's an enzyme. A catalyst is a substance that speeds up chemical reactions in a system without actually being used up in it. All right. So proteins serve as catalysts in the human body, and these catalysts are called enzymes. Okay. In other words, enzymes are biocatalysts. Uh, Catalysts in living systems that speed up metabolism, speed up chemical reactions. Proteins also act in defense. How's that? Immunoglobulins. Your antibodies are nothing but proteins, and they fight off infections. They aid in transport, like proteins can attach to lipids at times, forming lipoproteins. That's how fats are transported in your body. Most of your hormones, like testosterone and estrogen, are also uh, carried throughout your body by being picked up by proteins, by being attached to proteins here, okay? They contribute to structural support, like your nails, hair, skin, all of that contains protein, right? They cause, let's see, what have we, they perform regulation, okay? Regulation of your cell activity. They cause movement, like muscles. Muscles are protein, and muscles enable you to move. They provide storage. Storage for what? Storage for energy, storage for calcium ions, which are stored in the muscles themselves, all kinds of functions there, right? Okay, so the bonds between amino acids are called peptide bonds, as you can see here, right? So here are peptide bonds. These are amino acids which are joined together, okay? Why are they called amino acids? Because they have an amine group at one end, NH2, NH2, that's amine group, and a COOH group, or the carboxyl group at the other end, which is acid. So this is the amine group or amino part, and this is the acid or the acidic part. So therefore, amino acid. So you have to identify these two structures and you'll know you're looking at an amino acid which forms a protein, okay? So what are the monomers of proteins? Amino acids. The name of the bond between them, peptide bonds, okay? So there's all types of amino acids, non-polar ones, polar ones, right? Polar means they have a slightly negative and a slightly positive charge. Non-polar ones have a uniform distribution of charge. Charged amino acids. So here in this picture, you can take a look at many different types. These are examples of non-polar amino acids. These are polar amino acids. These ones are charged. These ones are the ones which have some special functions. All right, so just for your review here. So protein conformation. Protein is made up of four different levels of organization. The first one is called primary structure. And it's very simple. It's like a necklace, beads on a necklace, right? It's a linear sequence of amino acids. All these amino acids joined together like beads on a necklace. That is how the primary structure of a protein is arranged, okay? So then comes the secondary structure. That's the second layer of arrangement. What is the secondary structure? You can either have this twirly double, almost like a helical structure called the alpha helix, or you can have the zigzag type of structure called the beta sheet. So these two are types of secondary structures, all right? So when your protein is arranged in a alpha helix or a spiral coil shape, that type of protein you'll find in your skin and hair, as you can see, and it gives elasticity to your skin and hair. Some examples of places where you have this type of a conformation for protein, like the beta sheet, in your enzymes, okay? Your enzymes have your proteins arranged in this form. So then is the tertiary structure. So what is the tertiary structure then? It again comes in two types, a globular protein and a fibrous protein. Globular proteins are like globules, all forming these spherical structures, and fibrous proteins are like fibers, as you can see here, right? Um, so this is the tertiary arrangement of protein. Finally, you have the quaternary structure, the fourth level, and that is when two or more protein chains basically coil up together, like you see here. So here you have four globular proteins. You're looking at hemoglobin, by the way, here. Hemoglobin is the protein that makes your blood red and carries oxygen. It's like in this four globular shape, all right? And then you can also have a three fibrous protein like this, like almost like a rope. So this is the quaternary structure of how a protein is arranged, okay? So primary, secondary, tertiary, and quaternary levels. There's also prosthetic groups, which means some other non-protein groups which can be attached to the protein itself. Denaturation, what does that mean? When you heat up protein to a high degree or freeze it, it loses its function. And that's why high fever is never a good thing. If you have high fevers, temperatures in excess of 100 degrees Fahrenheit for long periods of time, then your body's enzymes get denatured. They stop functioning well, and that's when you develop metabolic failure and problems, right? So if a person has consistently elevated body temperature, do everything you can to bring the temperature down, whether it needs uh, Tylenol or cold sponging or whatever, right? Because this denaturation is usually irreversible. So, and it could be either because of an increase in temperature or because of a change in pH. If your body uh, acidity or alkalinity goes up or down too much, or your temperature increases too much or decreases too much, that will cause denaturation of your enzymes, all right? Uh, So pH changes, right? Now, think of some conditions that can cause uh, your body to become too acidic. The number one cause is seen in diabetics who do not use insulin or skip their insulin shots, and that is called diabetic ketoacidosis or DKA, diabetic ketoacidosis, all right? In the lack of insulin, since insulin is needed to push sugar inside of your cells, and if you are lacking insulin, so the sugar is not getting inside of your cells. It's swimming all over your body, but not getting inside of your cells. So this is a paradox. Your body is swimming, is thick with sugar, but this sugar is not being utilized, unable to enter your cells to be used for energy. So then what do your cells do? Then your cells basically start burning fat in order to get their energy. But when you burn fat to get energy, uh, a product of fat burning is something called ketones, and ketones are acidic. So in the lack of insulin, you become ketotic or acidotic, diabetic ketoacidosis. So a fair number of patients that you will see uh, coming in with diabetic coma, that's what they have, right? Diabetic ketoacidosis. They skipped their insulin shots or whatever, and so they had too many ketones in the body, and these ketones are acidic. They mess up their pH. The pH goes too down. They become too acidic, and then they develop coma and seizures and all those kinds of things. So you have to correct the situation. How would you treat DKA? Give them insulin first of all, and keep them hydrated. Dehydration is the other major risk factor here. So hydration, right? IV hydration, because obviously they cannot drink, they're in a coma, and sliding scale insulin. This is how you would treat someone who is in diabetic ketoacidosis. Some other examples or causes of acidosis in humans, sleep apnea. Sleep apnea, which is way more common in people who are overweight and not physically active, right? They have a thicker tongue, a thick neck, and so they stop breathing and they snore loudly usually when they sleep. They literally stop breathing for seconds, sometimes 2 minutes, and then they start snorting and sleeping. Breathing again. Sometimes they don't never wake up because sleep apnea could prove fatal. And so what happens is their carbon dioxide levels are perpetually elevated because they're not breathing properly. Carbon dioxide is staying in the system. That can give you acidosis as well, right? So some causes of acidosis, diabetic ketoacidosis, and sleep apnea, all right? How about alkalosis? So some causes of alkalosis are protracted vomiting, okay? Such as someone who's pregnant, and pregnancy might cause protracted vomiting. This protracted these episodes, right? And when you vomit, you're getting rid of your stomach acids, right? And so when you get rid of your stomach acids, you become alkalotic. Acid is gone, you become alkalotic. Also, anxiety and panic attacks. What happens in anxiety and panic attacks is, again, you breathe too fast, right? Hyperventilation. So you blow off extra carbon dioxide, you become alkalotic because carbon dioxide is acidic actually, okay? So some causes of alkalosis and acidosis and how to treat them is something that we need to keep in mind. So what distinguishes the tertiary and quaternary levels of organization? In tertiary organization, you have either like a like a three-dimensional structure, or rather a two-dimensional structure, right? Either like a globular protein or a fibrous protein. In quaternary level, you have a three-dimensional structure, right? Like what you find in hemoglobin or some other kind of fibrous protein. One is 2D, one is 3D. What happens to a protein when it denatures? It stops functioning, it changes its shape. How does a protein denature when it is exposed to higher than normal hydrogen concentration? Because that's too much acid, right? High hydrogen ion concentration means low pH, which means acid, and acid messes up the shape of the protein. It loses its structural integrity, its structural shape. All right, so that was it. We are done with chapter 2. So again, remember to keep up with your lab work that I'm sending out. Do as much as you can, right? Don't stress out over it. I will grade that over the weekend. I will be sending out your study guide for your first lecture exam. That is on Tuesday. So use it to study for that. I will send out all the instructions. Any questions, feedback, suggestions that you might have, do not hesitate to email me and let me know. Uh, enjoy the rest of your uh, evening, and I'll see you all tomorrow virtually again via recorded lectures. So have a great day. Bye-bye.